All right, now we're going to do sense organs. When you're talking about sense organs, you're talking about nerve endings, so we're going to, because they're going to be receiving information. So these are somatic, general, or cutaneous, various names for them. But really what it's doing is these are receptors. So it's anything that can detect a stimulus is a sense organ. All right. Most of them are covered with connective tissue or epithelium or something around them that will respond to that stimuli. The types of these, our main types are chemoreceptors. So these are going to have a chemical reception of some kind. Smell, taste, these are chemical receptors. Thermoreceptors, we're talking about detecting heat, hot or cold. Nociceptors are talking about sensing pain. Mechanoreceptors are movements and positioning, and then photoreceptors are light, so those would be your eyes. Distribution of these things. Most of these they're all over the place. Okay, and then we have some that are specialized that we find for our eyes, our ears, our nose for smell, and our tongue for taste. Those are our special ones. And these are innervated by our cranial nerves. All right? The others are, again, the somatic ones because they're all over the place. We're at someone's body, so they're going to be all over the body. We have interoceptors, excuse me. So these are ones that are going to be internal organs. That's what the visceral is. These will detect if our stomach is full, so it's stretched. That's what we're talking about right here. The stretching of our internal organ, nauseated, any kind of pressure on them. So those are, but again, these are internal. Proprioceptors are with our joints, our muscles, because these are our tendons and ligaments for that matter, because these are going to tell us the positioning and the movements of our appendages. And then we have our exteroceptors. These are going to be in our skin, again, eyes, ears, nose, mouth, for receiving anything coming from the outside. So intero, extero, and then our proprioceptors. Now, of these nerves that we find in here, we have ones that are called unencapsulated. Unencapsulated means that there's not going to be any kind of connective tissue wrapping around them. These are mainly there to detect warmth, so it's warmer or cold, and pain. Pain is a good thing. These are tactile discs, formerly known as Merkel discs, and these are just the little flattened out areas at the end of them. You can find these definitely on hair, where they call them peritrichal endings, and this is what hair is for monitoring movements of the hair, which tells us if there's an insect or something crawling on us. Again, these are unencapsulated because there's nothing surrounding them. Our encapsulated receptors, again, connective tissue or glial cells surrounding them. We have several of these. We have our tactile ones, which we find in our fingers a lot, because these are for touch and for texture. Ruffini corpuscles, again, movements, lots of pressure on them, not the soft, delicate stuff. Lamellated corpuscles, these are deep pressure, so these are going to be below the cutaneous region, again, for stretching, tickling. We got our bones, our viscera, which are our organs inside us. We have, excuse me. And we have our muscle spindles. These are going to, as muscles, these are telling you if our muscles are being stretched out, or which is going to trigger that reflex. If you go back a few lectures, the reflex of your knee kicking, because when you tap the tendons there, tendons, it stretches them and tells it the reflex. Again, that's the same with the next one, the tendon organ. And also with the joint kinetic receptors. Those last three are fairly similar to each other. So just be familiarized with these. From your textbook, chapter 13, shows you some of those unencapsulated corpus nerve endings. So they're free, again, body tissues, where they're located at, what they do. And then we have our encapsulated ones. So you see the connective tissue around them, that are the glial cells, the myelination going on them faster because they are myelinated. Again, body location and what they're sensing. Okay, so there's charts, tables in your textbook to review these if you need to. A receptive field. This is the size of area that a nerve ending receives information from. Now on like your fingers, the nerves are really close together, so they have a really finite or a fine sensing ability, so you can sense the texture or the shape of things with your fingers. But let's say on your back, where you don't need to know that, you'll have one nerve that'll stretch over a large area. So if you get touched in one spot, you might think it's a little bit different area, so it's kind of hard to locate the area because, again, it's one nerve covering a larger area. That's what the receptive field is talking about. So places where we need to have lots of definition from it, we have a narrow receptive field. Fingers are a good place for that. So again, anywhere in that field that is stimulated is coming from that field. So again, that's what I was talking about. If you have a spot on your back, it's like, it's right over here. And like, well, no, it's not. You got to get a little bit further because it's a larger receptive field, but we can feel it really easily with our fingers. And that's a smaller receptive field. Now coming back to that pain. Again, pain is good because it keeps us from injuring ourselves. All right. Again, it lets us know, whoa, hey, I got to stop doing that, whatever it is we're doing, or not do it. It is not the overstimulation. We have certain nerve endings that are specified for this purpose. All right, so they're doing their job. A couple of these, these are going to be myelinated, all right, so they can go fast. Most of them. So if it is a sharp pain, this is a highly myelinated nerve, and if it's a slower one, then excuse me. Or if it's a dull pain, I mean, it's a slower one, it just takes a little bit longer to get there. So we have both types in regions, right? And pain is very difficult to diagnose because pain will occur in different areas, depending on what it is. Where do people think of, okay, if you're having a heart attack, where do you go? Pain in your left arm, but your heart's over here. Different areas. So that's just one reason. That's one way to give you a good example of what it is. So here, this is what we're talking about. This is referred pain. So they're showing you if you're having a heart problem, it could be up in your neck, your jaw, all the way down to your pinky for the heart. Look at your gallbladder. It could be your right shoulder. Could be below your liver over here. It's connected to the liver. Kidneys could be your legs. All right. So these are typical referred areas for specific or for certain organs that people, the doctors can really, if you're having this pain in certain areas, like, okay, it's probably this, we'll start looking for that. But this gives them the idea and gets them going in the proper direction. This is referred pain. And this is why, again, pain is difficult to locate, especially the internal organs. All right, special senses. These are pretty much all located in our head, close to our brain. Our chemical ones are taste and smell. We have hearing and movement with our ears, and sight with our eyes. Start with our taste. There's approximately 4,000 taste buds on your tongue. And these get broken down into different types and different regions. So we have the filiform papillae, little bumps on the tongue, gives it the texture of it. You think of a cat, or you have a cat that's never licked you. And these are not taste buds, though. Okay, these are just friction for controlling what you're eating. The foliate papillae, on the size of your tongue, not well developed in the humans. I'm not worried too much about it. The fungiform papillae, these are mushroom-shaped ones, hence fungi. These are the tip of your tongue, which is why you go just touch your tongue to something to get a flavor of it. So these are sensing certain things. Then you have your vale papillae. These are near the back of the tongue, and there's about half of your taste buds are back here. And I'll give you an example of like a sommelier, a wine taster. When they sip wine, they let it get on the tip of their tongue, and then they'll let it roll to the back of their tongue because you get different flavors that way. So if and when you, if you're old enough, then try it if you want to. If you're not, wait till you're older. It's not that big a deal. So they're showing them here. There's those, not the full those on the side, the vale papillae right here. They're showing right here inside these little things on the side of them here. This is where the taste buds are in this region. This is an actual taste bud, a magnified view of it. Okay. Your fungiform ones, these are all, there's a lot of them at the tip of your tongue, but there are all kind of all those ones that look like kind of mushrooms that are taste buds also. But you have larger ones back here at the back of your tongue. All right, smell, again, is going to occur in the olfactory region of the brain. So we have about 10 to 20 million of these neurons that are doing this. The hairs in our nose catch our odor molecules and then transmit those to the nerves and so forth, which takes it to the olfactory lobe, which is going to go to the brain, which lets us identify a sense of smell. As you see, comes in, then you have the nerve endings hanging down, and that's where it's going to go stimulate these neurons, and those are going to send the impulses to the olfactory bulb or tract. This is the olfactory bulb, and take that to the brain so that your brain can identify this. Your sense of smell is located very close to your memories. And so your sense of smell will actually trigger memories better than sight or sound because of that. That's why a lot of people smell cookies and think of their grandmother's house or something, because your grandparents were like always making cookies for you when you were a little kid or something. These are stereotypical type things that happen. Your ear. You have three parts, your outer, middle, and inner ear. These are for, your ears are about hearing, your balance, and a few other functions go in there, but for your coordination on it. So the outer ear, the pinnae, auricle, just help focus and everything. There's hairs inside of it. We also secrete earwax, which is called cerumen, and this prevents infections from happening. I guess not 100%, nothing is, that's why we get infections, but it prevents things from, it makes it an inhospitable environment for bacteria, fungi, or even insects to get into your ear. Not saying it can't happen, but it reduces the likelihood of it happening. The middle ear is going to consist of the tympanic membrane, the eardrum, the eustachian tube, which is a tube from it that's going to carry it's easier to picture, and then the bones of the ear, the ossicles, known as the malleus, incus, and stapes. The common terms are the hammer, anvil, and stirrup. The inner ear then is the cochlea and the semicircle canals. Now we're going to kind of go over all these. Remember, cochlea is always going to be, it's snail-shaped thing, so it's easy to identify that way. And the semicircular canal, these are for balance and coordination. So again, here's all the outer ear, okay? We go to the inner ear, starting with the tympanic membrane. There's the eustachian tubes. There's your the three bones of the ear. There's the cochlea, and these here are your semicircular canals. Again, tympanic membrane vibrates from the sound. It's going to move the bones. Bones are going to come over here and affect your semicircular canals and the cochlea, which are going to transmit impulses along the cochlear nerve, which is going to producing the sound. Here's your semicircular canals. These are, notice that we have them in three directions for your X, Y, and Z of your positioning, because that's how we know we're spinning or whenever our ups, down, left, and right. In these, so we come back up over here. So we're talking about these right here, the maculae, and then you have your cuticle and the seculae. These lets us know which way is up and down, because in here they have stones called otoliths right there, that detect the pull of gravity for us. And again, you see right here, someone thinking of a string on a ball on a string. If you hold the string, the ball falls down and it'll move around. Same thing. That's kind of what it does. And as you move your head one way or another, those otoliths move with it. It tells you what how, if you're upside down, left, right, or whatever. Your semicircular ducts, and again, these are going to give you your 3 directions. There's a fluid inside them. So if you're spinning to your right, they actually appear to be going to the left. Actually, if I spin to your left, they go to the opposite direction because of inertia. The fluid doesn't move as quickly. So when you're spinning and when you stop, it feels like you're still going. That's because that fluid is still moving. That's the dizziness of it. So again, let's come back. Here's those otoliths. As you tilt, you tip your head, they move. So it gives you your up, down, left, and right. There's your semicircular canals. They're talking about it right here. And they move from the fluid that's in there. And that's going to, so it's going one way. As soon as you stop, it's going to appear back the other way. And that's that dizziness effect that's going on. Then your eye, photoreceptors. We're going to really just know the parts of the eye. So in here, you got your lens, you got your cornea, the pupil, you got your optic nerve, you got the sclera, containing all of it. The fluid in it is called the vitreous humor. Yeah, those are the big ones for it. For people who are nearsighted, then that means that's called myopia. Their lenses shape different, so it causes them to have the image appear in front of it. So we get glasses to fix it and put it back. And hyperopia, which is just the opposite of it, is farsighted. These are people who can see things far away but not things up close. It produces it where it puts the image too far away, so we fix these with glasses. Some of the big things that we find in the retina, the back of the eye, we'll come back here, sorry, right through here, the yellow portion, are the rods and cones. And we call them that because they are kind of shaped like a rod or a little pointed here like a cone. Rods are for night vision. They don't really detect a lot of color. So they're the night vision, and the cones are for detect color. So whenever we're looking at an organ from some other animal, we can look and see if they have a bunch of cones, and we know they can see lots and lots of colors. And if they don't have a lot of cones, it's mainly just rods, that means they're basically for night vision and they don't see color. That's why we can say like dogs are colorblind, because they don't have a lot of cones, because they don't need to. They have a very good sense of smell and they can follow things that way. It's just an example. That is it for the special senses. Again, familiarize yourself with the major parts that we kind of went over, the differences between an unencapsulated neuron and encapsulated neuron, your chemoreceptors, thermoreceptors, nociceptors, mechanoreceptors, those things. Familiarize yourself with those. Be ready for the quiz.